Hi everyone, my name is Devan O'Keefe, and my research examines how maternal vitamin B12 deficiency disrupts myelination during brain development. Vitamin B12 plays a critical role in one-carbon metabolism, which regulates DNA synthesis and methylation, processes essential for maintaining normal cellular function and gene expression. When B12 is deficient, these pathways become disrupted, leading to measurable metabolic changes and, importantly, potential neurological consequences. At the center of this disruption are two key molecules, methylmalonic acid and homocysteine. As shown in Figure 1, both accumulate when B12-dependent metabolism is impaired. Homocysteine reflects reduced methylation capacity, meaning cells are less able to properly regulate gene activity. At the same time, methylmalonic acid is directly neurotoxic and can cross into the brain. Together, these changes not only disrupt DNA synthesis, but also interfere with the production of myelin, the insulating structure required for efficient signaling in the brain. This leads to a central question driving this study. Does maternal vitamin B12 deficiency directly impair myelination during development? To address this, we used a maternal diet mouse model, where dams were either fed a controlled diet or a B12 deficient diet, and offspring were analyzed at 11 weeks of age. We first needed to confirm that this deficiency was functionally present. As shown in Figure 2, both MMA and homocysteine were significantly elevated in deficient offspring, confirming disruption of one-carbon metabolism. But these metabolic changes do not occur in isolation. They have broader physiological consequences. We observed that B12 deficient offspring exhibited reduced body weight and shorter body length, indicating impaired overall growth and development. This suggests that maternal deficiency affects not only biochemical pathways, but whole organism outcomes. At the same time, we identified clear hematologic effects. Deficient offspring showed fewer red blood cells and larger immature cells, a pattern consistent with megaloblastic anemia, reflecting impaired DNA synthesis and abnormal cell production. Together, these findings demonstrate that maternal B12 deficiency produces systemic effects across multiple biological levels. However, the most critical question is whether these disruptions extend to the brain. To address this, we examined gene expression in cerebral white matter, focusing on pathways involved in oligodendrocyte development and myelination. Our findings reveal a very specific pattern. MBP expression is significantly reduced, indicating impaired myelin formation. SOX10 shows sex-specific changes, with greater effects observed in females. In contrast, OLIG2 and PLP1 remain unchanged, suggesting that early oligodendrocyte development is preserved, while later stages of maturation are disrupted. This distinction is critical. It shows that B12 deficiency does not only prevent these cells from forming, but instead impairs their ability to mature and produce functional myelin. This provides a clear mechanistic link between maternal nutrition and disrupted brain development. More broadly, these findings extend beyond developmental biology. Disruption of myelin is a central feature of diseases such as multiple sclerosis, and growing evidence suggests that myelin dysfunction may also contribute to neurodegenerative diseases such as Alzheimer's. Our results suggest that impairment of vitamin B12 dependent pathways may represent an underlying mechanism contributing to these processes by limiting proper myelin formation. The key takeaway is this. Maternal vitamin B12 deficiency impairs myelination by disrupting oligodendrocyte maturation, linking nutritional deficiency to long-term neurological risk. This work highlights the importance of adequate maternal B12 intake, particularly in at-risk populations, and demonstrates how nutritional status can directly influence brain development at the molecular level. I would like to thank the Erickson Discovery Grant, which I was awarded in 2025 through Penn State, for supporting this research, as well as Dr. Rita Castro and my research team. Thank you for your time and for listening.